So we are going to discuss about the topic of intestinal schistosomosis. I am Major Dr. Krishnendu Kundu, Assistant Professor from the Department of Veterinary Parasitology, Faculty of Veterinary Animal Science, Institute of Agriculture Sciences, Banaras Hindu University. So first let us know what is schistosomosis and what is intestinal schistosomosis. So intestinal schistosomosis is a disease of ruminants which is caused mainly by the genus species from the genus Schistosoma. This includes several genera like Schistosoma indicum, Schistosoma bovis, which are the most common parasites, trematode parasites of domestic ruminants in India. Now, we will mostly lay emphasis on Schistosoma indicum since it is a common cystosome of the parasites in India, cystosome of ruminants in India, which is responsible for intestinal schistosomosis. Now, the cystosomes, they come under the family Schistosomatidae. As you can see, they come under the family Schistosomatidae, and they are also called the blood flukes. Blood flukes, why? Because these trematodes, also known as the flukes in common language or in a layman term, they are found in the blood vessels of the tissues. For example, in case of Schistosoma indicum, the adult stages of the parasite, they will be found in the mesenteric veins as well as in the hepatic veins. Now, if you see these cystosomes, unlike the other trematodes, the cystosomes are typically bisexual. They have got separate male and female organisms, unlike the other cystosomes, unlike the other trematodes like the fasciola, which is hermaphrodite. So here, this is electron micrograph structure, is you can see in the photograph, which is of Schistosoma indicum, and this large structure, as you can see here, you can find what you see is the it is a male. So this is one oral sucker and this is a ventral sucker, and here inside this is a canal like structure in the male. This particular structure is called the gynecophoric canal. So inside the gynecophoric canal lies the female cystosomes. Now, sexual dimorphism is well marked in this particular genus. The adult female is carried in the gynecophoric canal of the male, and unlike the other trematodes, the number of testes. So, in case of trematodes, there are only a pair of testes, but here, in case of cystosomes, they have multiple testes. Okay. Coming to the general morphological characters, apart from the sexual dimorphism, what you can see, so this is another electron micrograph structure, micrograph, which shows the spine-like structures on the body of the cystosome. So here you can see the small spines. Now the cystosomes, they have got a oral sucker, as you can see it here. This is the oral sucker. There is a ventral sucker, okay, and same it is for the female as well. There is a oral sucker and there is a ventral sucker. Now fertilization takes place. There is an intestine. So from the oral sucker comes the pharynx, and then there is an intestinal ceca. The intestinal ceca, they branch. So you branch and they again reunite and they again rebranch. So you can see there is again a rebranching. Okay. So this is the general morphology. As the testes, they vary in numbers. They may be 3 to 5 or 6. And in case of females, there is one ovary, as you can see it here. And fine. This is what is the life cycle pattern of the cystosomes. So cystosomes, the eggs are laid near the water bodies. The eggs come out through the feces. The eggs contain an embryo. It immediately hatches miracidium, and this miracidium in water, it can survive for 24 hours. It will look for a snail host. So once it enters into the snail, it undergoes development, and this cercaria is released from the snail. Now this cercaria is the infective stage for the ruminants. So as you can see, the eggs always have a spine. So this is a spine, as you can see, sharp spine. which is typical of cystosome eggs, and cystosomes can be identified on the basis of that spine. So coming to the summary, so these are blood flukes which are found in the blood vessels of veins of the mammalian host, specifically the mesenteric veins, and sometimes even in the hepatic veins. The eggs are already embryonated when laid. They contain a miracidium. The eggs are the most pathogenic stage. So whatever disease you find, that is enteritis or hepatitis, it is mainly because of the eggs. The egg antigens are responsible for the clinical disease. The eggs hatch in water when they come in contact with water, and the miracidium is released. The miracidium infects the snail host and the cercaria, and the miracidium undergoes development in the snail, forming the sporocyst and ultimately the cercaria, and this cercaria released from the snails. And unlike in case of other trematodes, this cercaria do not insist to form a metacercaria, but rather they infect the host through skin penetration. Okay, thank you.